targeted by an addictive and therapeutic psychostimulants, including cocaine and amphetamines. Proteins termed neurotransmitter transporters. Dr. Blakeley's work has uncovered mutations in genes encoding these proteins, work that has shed light on mechanisms that drive cardiovascular, neuromuscular, cognitive, and behavioral disorders such as ADHD and autism.
Blakely?
Thanks for I guess I will continue a theme that Richard Fry noted of pearls and following data in a career where a certain trajectory is charted. You can still follow that trajectory, but in the meantime, you see these things and they look really interesting. Things are going well. If things are grabbing well, grab a few of those, and then sometimes it actually takes over. We're going to talk about serotonin, autism, and the immune system. We've heard about all of those things at this meeting already, in one form or another, and I'll tell you how we got into this. So for those who are not schooled in neuroscience and neurochemistry, so forth. Great. So we'll focus on the serotonin system. It's wide-ranging, monoamine neurotransmitter system that projects widely throughout the brain, modulates a lot of key functions that we know are disrupted in autism, social behavior, repetitive behavior, and communication. And it's a very simple molecule, synthesized from tryptophan, and released at synapses, axonal veracosities, as well as a few classical synapses. Now, when I got started in neuroscience, I learned about transporter protein. I learned about transporters as these inactivating proteins that would clear neurotransmitters from synapses. What I was taught was that there were these blobs here, these green blobs, and this is what was shown to me in grad school. Somehow these miraculously clear neurotransmitters out of the synapse and kept the activity at receptors low so that you could see a pulse coming the next time. And I got fascinated with this and learning about psychotropic drugs and then Prozac that could block these proteins and elevate amines and impact physiology. So I wanted to get a little bit deeper into what those green blobs were. And was fortunate enough working with very talented postdoc Amanda Ramamurthy, human serotonin transporter. 30 years ago, almost to the day, we reported this in this paper in PNAS, and wrote versions of that earlier, other family members like norepinephrine transporter and its various dopamine. And in the early 90s, this field exploded with lots of these proteins with sequences. It would take another 20 years before we would ever get anything close to any high-resolution structure. So we had to do a lot of hand-waving still on how these drugs were interacting with the protein and how the protein was being controlled. But finding the human serotonin transporter gene located in right here by then the ancient technique in situ hybridization. We were so excited that we had this gene, and this was an antidepressant target, and surely this would be the gene that would hold all the clues to depression, right? Well, not so much, but it's driven a lot of pharmacology, and our clones went off and were used for drug discovery, and we studied the regulation of this gene, and I'll show you some on the protein, but depression took a little bit of a back seat, as I'll show you going forward here. And that was generated by this finding when we were doing some studies, not expecting to see the serotonin transporter this early. This is a mouse embryo, mid-gestation, E13. And these neurons that are in the midbrain and brainstem here are the ones that are serotonergic, and they're roaring with expression of the serotonin transporter. And by this time in development, there are fibers coming off of these cells moving into the forebrain, and the transport activity can actually be measured. And it's important. If you block this transporter or it's defective at this stage, you start to get problems with brain development. So it sort of shocked me a little bit, set me on a different path. which was to think about juvenile onset brain disorders as opposed to the adult ones that we usually think about in using SSRIs. But we kept going on the work we were doing, and but recognized that this was probably meaningful because others, here's a study that came out a little bit later from Pat Levitt's lab showing a distribution of serotonin receptors. This is just 2. You can go to the Bonin paper and see a whole bunch. But in blue, you can see here the density of different serotonin receptors in the mouse brain very early in development. And we now know they participate in different aspects of neuronal development. So after moving my lab, after cloning the serotonin transporter at Emory, I moved to Vanderbilt. And there I happened to have a great interaction with a geneticist named Jim Sutcliffe, who had been working on Angelman syndrome and was moving into autism. And one day on his students' thesis committee, he brought in some linkage plots. Linkage analysis isn't done much these days, but it's what gave us the mutations in CFTR and Huntington's disease, and it's a family-based approach to look at heritable changes in the genome by descent and looking at what's transmitted to affected versus unaffected offspring. And that's what Jim did, and his student in his meeting showed all the linkage plots in the different chromosomes, and there over chromosome 17, Q11.2 was a large linkage peak, and it was male specific. And we thought, you know, we should go look and see if there's anything in the serotonin transporter gene that we might be contributing to that linkage. And by that time, I had already started screening for human mutations in the serotonin transporter, and it was turning up a dry well. And that was just because we were looking at population samples of normal DNA, pretty much, normal individuals typically developing. And out of this, actually, we pulled out 5 coding variants. And 4 of these are private mutations, 3 of them private mutations, and 2 of them a little bit more abundant. And I'm going to talk, whoops, it actually does work. Okay. I'm going to be talking about this one on the N-terminus, this Gly56 alanine, and I'll show you why in a minute. But the frequency of these variants told us that this was more than just a random draw into the community. We would have never seen this many, and they all had a very interesting function, and that's here. Now we're plotting transport activity, in this case in transfected cells. But what you can see is compared to the wild type SERT, all of these have elevated levels of serotonin transport activity. So some of you may be thinking that all mutations kill proteins, or they disable trafficking to the surface because they're misfolded, something like that. In this case, there was more function evident, and we worked out the basis for this over time, and it converged with the other side of the lab that was doing the work to actually pay the bills, which was study how drugs interact with the transporter and how it was normally regulated. You'll see this convergence in just a second, right now. So in that effort, we had established a couple of signaling pathways that normally regulate the transporter. And there were 2, particularly, that we knew if we activated them, the transporter would gain activity. And we thought, well, is there a relationship between that signaling, which causes a gain of activity, and these mutations, which have a gain of activity without activating anything? And in these plots, showing you a demonstration that there is a connection. You can see all of the variants laid out here, transport activity going this way. Here's the wild type transporter, and what we're doing in these figures here is we're activating the PKG pathway that, via analog of cyclic GMP, and you can see stimulation of the transporter, and we can block that with antagonists. And then you can see that these 3 have a slightly blunted level of activity. The ALA56 variant, not responsive whatsoever, okay? And in the second pathway I told you was P38 MAP kinase. This is an activator of P38 MAP kinase. You can see stimulation. These 3 are a little bit less, and this one, no activity whatsoever. And to us, what this said was, this is particularly interesting because this has elevated activity and you can't stimulate it with PKG or P38 pathway. So that says that that mutation has put this transporter in a high activity state, probably by copying the PKG and the P38 stimulation. It's on that pathway and you can't get any further. So we set out to explore that a little bit. And we utilized the fact that in discovering this PKG and P38 pathway, we found that the PKG pathway has one step to shuttle serotonin transporters to the cell surface, and the P38 MAPK pathway is downstream of PKG, and it actually turns this transporter on once it gets to the surface. Now this will relax back to a less active state, and if you come in with another molecule that can activate P38 MAPK, such as through this immune receptor that you heard Ning Kwan talk about, the IL-1 receptor, you can turn it back on. So this was a discovery that these transporters are not just sitting there all the time taking up their neurotransmitter, they actually go into low activity and high activity states on the membrane surface without trafficking. And so that linked us now back to the mutation studies. This is a little of the basic science that went into it. Here you say IL-1 beta and TNF-alpha, which will do it as well, stimulating transport activity. In this case, this is a serotonergic culture preparation, and you see it as a function of concentration or time. Very nice stimulation. And then we took IL-1 beta and injected it into the hippocampus, and then we could monitor with voltammetry, with electrodes that can monitor the clearance of serotonin once you've injected it. And it takes a certain amount of time in the wild type, but with IL-1 beta, it takes much less time. It speeds up the transport activity of the transporter, just like it did in vitro. Now that was now starting to converge with this gain of function work that we had just come out of with these mutations. And when I started to think about what the pathways were that might be connected to PKG and P38, and even before the IL-1 studies came along, we had these findings by one of our colleagues at Vanderbilt Karolyi-Murnix on a post-mortem transcriptome study. And what they found was that the major pathways, among the major pathways that were elevated in the temporal cortex of subjects with autism, was the IL-1 receptor and P38 MAPK pathways. So this said, okay, there's more here to it than just these sort of basic science studies of regulation. There's something that's probably disease relevant, and we've already got a toe in the water. Okay, so if they're connected, if the immune activation of the transporter happens in vivo, we should see IL-1 receptors on serotonin neurons. And up to that time, it wasn't possible really to get good visualization of the IL-1 receptors expression, or to, because the antibodies were not very good, and we're just starting to get into fluorescent in situ hybridization. And this is some work from Paula Gajewski-Kurtzel in my group, and also from Ning Kwan's group with a transgenic mouse. And in this particular case, this is a mouse that is expressing the IL-1 receptor. So it is using the IL-1 receptor promoter to drive fluorescent reporter protein, and we could blot for that, or we could stain for that. And what you can see is this is the serotonin neurons in the midbrain, and this is the serotonin stain, highlighting all of them. And you can see the IL-1 receptor here, very strongly expressed in this dorsal group. You see less down here, but if you crank up the volume, you'll see it's expressing. It's just not at the level that these are. And here's the merged image. So this is an indication that at least the promoter is working and expressing in those cells. And I'll show you better evidence. So if it's expressing the receptor in the serotonin neurons, then somehow the cytokine, the inflammatory cytokine, should do something to serotonin neurons. So Hideki Iwamoto in my lab has done slice recordings, and you can see here in patch-clamped serotonin neurons that are identified fluorescently with appropriate markers, you can see that IL-1 beta will shut down a neuron that is tonically firing because we stimulate it, and the IL-1 beta will shut this down. And you can see statistical values here. We can block that with the IL-1 receptor antagonist. And then in the EPHIS analysis, what we see is that we've got a reversal potential and a property of this channel that looks like it's a potassium channel, inward rectifying potassium channel. Those are channels that are known to shut down serotonin neurons in response to 5-HT1A autoreceptors that normally keep the firing of these neurons at a fairly irregular pace. Here we're showing that an inflammatory stimulus can turn those neurons down too. Now you remember the P38 MAP kinase, and I was illustrating IL-1 receptor goes through the P38 MAPK, and that turns the transporters on. So we said, okay, well, let's put the IL-1 beta on and let's inhibit P38 MAP kinase once we have it inhibited. And you can see the turn down here, and now we throw in the P38 MAPK inhibitor and it comes back. You can see that nicely here. So that tells us we've got a pathway. We don't know all the elements in it, but at least it's using those signaling enzymes that we had seen back in the original studies that variant could not respond to. So that's pretty interesting. So here we show IL-1 beta stimulates serotonin, it inhibits serotonin neurons through a P38 MAPK pathway, and the G56A variant is already elevated and doesn't respond further to PKG or P38. All right, so we then said, okay, well, this is all fun, playing around with kinases and applied peptides, and let's see if we can do something in vivo. So how do we activate quickly IL-1 beta in the brain? Well, the way that most people do it is to inject an animal with LPS or poly-IC, something like that. So we did that, and this is an in vivo amperometry experiment. I showed you that when we injected IL-1 beta and looked at clearance. Now what we're going to do is inject LPS and monitor clearance of serotonin. So we're going to puff, 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 puff over time, and we're going to watch the clearance curves as they take away serotonin each time. And so you can see here, pre-LPS, you see we puff serotonin, you can see the clearance rate here. And then as time goes on, and here we're at, I think this is 65 minutes. I can't actually see it, but it's something like that. And you see that this clearance is becoming steeper and steeper. So this shows us that LPS, which will generate a cytokine storm and cause an increase in IL-1 beta and some other cytokines, will cause in vivo this transporter to kick into high gear. Okay, so now if that's true, that happens, and that's done in an anesthetized animal. What if we inject LPS into a living animal that's walking around and monitor activity in vivo using the poor man's way, using CFOS activation? All right, now what you can see here, this green is males, and this pinkish orange here, the color scheme, females. And this is the dorsal raphe here. And what you see is that we see an inhibition of activity with the LPS. And if we knock out with a floxed IL-1 receptor, the IL-1 receptor just in serotonin neurons, you see we don't see that inhibition anymore. Now that's pretty interesting because we only knocked out the IL-1 receptor. But TNF-alpha will inhibit these neurons. There's TNF-alpha receptors on these neurons. But somehow in this particular condition with this LPS, IL-1 beta is dominant on this, in this effect. Okay, so then the question becomes, where do those neurons go, and does that transmit a signal someplace else? So doing the same manipulation, we inject LPS, we know the serotonin neurons are getting inhibited. What happens downstream? And the consequences there depend on sex. So on the left side, this is a male and this is females. And what we see and I'm just showing you one experiment here, This is in projections to the lateral habinula. And in the serotonergic projections to the lateral habinula, you see this CFOS activation occurs in the Cre minus line, and then when we have Cre plus, it goes away. Okay, so the serotonin neuron expressing IL-1 receptors are needed in order to get that stimulation in the lateral habinula. And over in the females, the lateral habinula is inhibited with CFOS, and you can normalize it by getting rid of the receptor. So this particular projection has a consequence that is sex dependent. So if you have an inflammatory insult, the readout in the lateral habinula is going to be opposite. And that circuit will drive a number of negative affective states. And we are working now on aspects of projections of the lateral habinula to social behavior sites, to repetitive behavior sites, other mood-related sites, and so forth. So that's work that's going on now. Okay, so when I was showing you the model, we were talking about the transporter and its regulation through those pathways. And we historically, and we think about the transporter being on the axons, not on the cell bodies where all this firing is going on and transmitting reduced firing out to the processes. So what about the axons? And here again, we take advantage of Ning Kwan's mouse, not because it has a transcription reporter, but because it brought an HA tag with the IL-1 receptor when you restore it back in serotonin neurons. Well, that was a godsend because the protein antibodies just don't work. But here comes that HA tag, and we can now do this experiment where we stain for SERT in the forebrain. This happens to just be in the hippocampus, and I hope you can see this. It looks bleached out a little bit from my side, but here's the IL-1 receptor with that HA tag, and if you can see, like for example, this little process here, and you see it here with the tag, and now here's the overlap looking at the dual labeling. And you can see beautifully that at these, you see these little puncta going along here? That's your varicosities where serotonin is being released and where we're getting our transporter expressed. And then right there is the IL-1 receptor ready to regulate the terminals as well. So there's 2 points of regulation on those neurons. Change the firing, increase the transport. Both of those together will drop the serotonin levels, and we know that happens from other studies. Okay, can we see that biochemically? If we make synaptosomes, which is the nerve terminal preparation from the brain, in this case it's from the midbrain, take those synaptosomes and we add, we've injected LPS, we made synaptosomes after that injection. We see the stimulation of SERT activity in the terminal preparation. And if we knock out the IL-1 receptor from serotonin neurons, you see it's gone. So this is evidence now that this IL-1 receptor and the serotonin transporter are in close coordination in terms of how the serotonin neurons respond to an inflammatory insult, at least acutely. Okay, all of these experiments are acute. They happen. The addition of IL-1 beta will happen. This will happen in minutes. If you have LPS, it'll happen in 20 minutes as you go in a little bit further. So our model then has been that IL-1 beta elevations that activate the IL-1 receptor can activate P38 MAP kinase and can cause, and we know this phosphorylation occurs, can cause a transition of the serotonin transporter from what we call SERT to SERT star. And SERT star is a higher activity to clear serotonin. We can block that back down with an SSRI. Obviously, we use those as antidepressants, and it raises the question then of, do we ever have ectopic SERT star? Does it happen without inflammation? Well, I gave you an example, and that's ALA56. That mutation puts it in this SERT star state. So we figured, okay, well, let's bite the bullet, and let's knock that mutation into mice, and let's see where that'll take us. And what we found when we did this, the first thing we did, and this is where Jeremy Vanderbilt right here will talk to you next, when he was a postdoc in the lab. And what he found was that there was elevated serotonin in the blood. which we believe is because of SERT being present on platelets, where the majority of serotonin in blood is stored. This is a major biomarker for a significant number, 25–30% of individuals with autism. In the brain of those mice, when we pulse serotonin, what you see in gray is the wild-type allele, and in blue is the mutant allele. And you can see that significantly faster clearance, which we saw over multiple animals, statistically significant. So the mutation is doing what we showed in vitro now in vivo. It's hyperphosphorylated compared to the wild type. We know that that's P38 MAPK dependent phosphorylation using inhibitors. If you cut slices from the rat and monitor firing of the serotonin neurons, again you see that they're inhibited as IL-1 beta was added. But there's no IL-1 beta here, so the mutation has already pushed the system into this hyperactive state, leading to reduced activity. Now, I don't have time to go through all the behavior experiments that were done, but I just want to show you this one because it probably connects the most visibly to thinking about autism. Jeremy found repetitive communication deficits as well. This is a tube test of, it's called a social dominance. I like to think about it as social avoidance. And you have two mice that enter at these different ends, one a wild type and one a mutant. And the question is who backs out first. And classically in the dominance literature, the one that backs out is called the loser. So please don't take this any further. This is not a good way of saying things. But regardless, what you see is that the alpha-56 mice back out in this interaction. So they have social avoidance. We've seen this in some other social behavior interaction tests as well. Alison Walsh, a graduate student, has a poster back there and has been working in that area too. So go see her poster and learn a little bit more about the alpha-56 mice. Okay, so is this a model that can be used to screen therapeutics? We've seen this a number of times in this meeting. Our work has gone toward this P38 MAP kinase, which appears to be overstimulating the mutant and being a critical player in the pathway by which inflammatory processes gain access to at least mood-regulated regulatory machinery of pathways, but also I think social behavior and other things. Marty Waterston's developed a very specific kinase inhibitor, MW150. It's specific because it picks out one isoform of P38 MAPK. There are 4 of them. The drugs that were available to this time were not brain penetrant or not isoform selective. This hits them both, and it's very specific for alpha, and the rest of these endpoints on this diagram are other kinases that doesn't touch. So that was great. We took that compound, and we gave it to this mouse. These are all studies in males. All the studies I've shown you up to this point, and will be, are in males. We gave this molecule once, and then we looked, and everything seemed normal. We gave it once a day for 5 days, and these are the data we got. SERT alpha-56 mouse, remember, clear serotonin faster. Now we had normalized it. It was now back to its normal clearance rate in the hippocampus. If you look at the tube test, this is what we had before. If we did this once a day for 5 days, now we go back, in this particular experiment, it looks like they're super winners. Doesn't hold up over a lot of experiments that way, but they do come back to being normal. And I should say as well, before we get to there, we also didn't want to just trust the pharmacology, so we knocked out P38 alpha MAPK in the serotonin neurons and repeated this. And you get the same result. So we believe it requires P38 alpha MAPK in serotonin neurons in order for this mutation to then trigger a physiological response in the serotonin neurons that leads to social avoidance. So we think that these are experiments that demonstrate the importance of this pathway and a genetic and pharmacological way to tamp it in. That compound MW150 right now is in trials in relation to inflammatory signaling in Alzheimer's disease, and we await some of those studies and would love to piggyback on it to try this compound out in various individuals with autism. So the last thing I want to point to and try to link this up with some of the talks that have come before is a model we're thinking a lot about right now. Has to do with the role that microglia play in homeostatically controlling the neuronal environment. And we generally think about this in terms of plasticity or in development and pruning, and we've seen a lot of the data on changes in neural complexity, dendritic complexity. And what we know is that these microglia in their homeostatic state carry serotonin receptors. And one of the aspects of clearing serotonin away a lot is to raise the opportunity for these microglia to become these microglia and become activated by other stimuli. Perhaps not by judgment cells, but other things that are happening in the environment. And so serotonin suppresses this and keeps it here. And so our idea is that when we have this pathway activated, not only are we changing serotonin dynamics in a neuronal context, but we're also likely to be changing the activity of microglia. And this is going to result in IL-1 beta, and we're going to get a positive feedback loop, and it's going to stabilize, which would predict that you would have a chronic inflammatory state after the IL-1 beta goes away, and you might have it with the mutation. So we've taken those mice and just done some PCR studies in different brain regions, and here you can see in the midbrain elevation of in the SERT IL-56 mice of IL-1 beta, IL-6, and probably a compensatory IL-10 expression elevation. So, I want to make a few points in closing, but first, just thank all the people here that have